Lower Blackwood Landcare Group. For those of you that don't know us, we are a not-for-profit, independent landcare organisation with an interest in sustainable agriculture and the broader environment and river health in the Lower Blackwood catchment. You can find out more about us and the work we do on our website at lowerblackwood.com.au. I'd also like to acknowledge the traditional owners of the land on which we all live and work. I acknowledge the Wadandi and Pibbleman people of the Bibbleman Nation and pay our respects to elders past, present and future. Thanks also to the Uptake Project for making this webinar possible. The Uptake Project is jointly funded through the Healthy Estuaries Initiative and the Australian Government's National Landcare Program. Now, before we get to our presenters, just a few words on how the webinar will run today. You may have noticed that by now you can hear us but not speak yourselves. That's deliberate so we can keep things moving along at a good pace. You can still definitely ask questions though. Just type your question at any time into the chat box and I'll make sure it gets asked at the end of the presentation. Also, if you run into any technical difficulties, please use the status button, the hand, which you see on the bottom right side of your screen to let me know, and I'll see if I can fix it from my end. Don't worry though, if you do have to leave early or drop out, as we'll be recording this session, which I'll email out to you next week. Lastly, your feedback is important. So if I can ask you to stay on for a few moments longer to complete our webinar evaluation form before you sign out, that would be great. And many thanks. Okay, to the reason that you have all tuned in, I've got great pleasure in introducing our presenters today. First up, we have Dr. Cassandra Schefe. Cassie, as she likes to be known, has over 20 years experience in soil chemistry and soil-plant interactions with a key interest in soil phosphorus. Cassie, are you there? If you'd like to turn on your mic and your video, that would be great so people can see you. After Cassie, we'll be hearing from the Department of Water and Environmental Regulations, Dr. Kath Lynch, who is the District Manager for Geograph's Capes District. And also Rob McFerrin, who is the Uptake Project Coordinator, with Graham Mussell assisting him. Graham Mussell is an independent agronomist who works with Rob. And then we'll finish off with some comments from two local farmers who have been participating in the project Uptake. So it's a pretty full schedule today, but we'll try and keep things moving along at a pretty good pace. So without much any further ado, I think I'll let you start. Over to you.
Thank you very much, Kate. It's great to be here this evening. I'm based in a little town called Rutherglen, near Albury-Wodonga in the eastern states. And apologies if there's any noise in the background. My family are just getting dinner organised on the other side of the door. So let's hope everything settles down. Okay, so let's talk about one of my favourite topics, which is soil phosphorus. And I know even though I come from the eastern states, I've had the benefit or the challenge of working with a lot of different soil types around the country, and there's certainly some key themes that come across regardless of what state you're in and what system you're working in. So today we'll go through some of the basic chemistry behind soil phosphorus and how the role of chemistry and microbes connect in. I've tried to break it down into things that make sense. If there's anything that you're not sure of, I'd love to explain it further at the end of the presentation. So basically today we'll work through why is phosphorus important, the P cycling in our soils, why is phosphorus availability an issue in terms of too little or too much. Can soil biology increase phosphorus availability or unlock phosphorus? That's something that I get asked a lot. Can we use microbes or our microbial community to increase our phosphorus supply? What else can we do to influence our Prelease? And generally, at the end of this, what can we do to move forward in understanding more about our phosphorus? So to break it down to the real basics in terms of P is it is necessary for all life. It's a key ingredient in our genetic molecules, in our DNA and RNA. It's required for cellular energy production, so ATP. It's a key component of our cell membranes, and we have a high phosphorus content in our bones, which was actually the early P source for fertiliser. So think of even our blood and bone type fertilisers, the phosphorus that's in them is because of the phosphorus structures in our bones. So basically, P is required for plant growth, and the more productive plant species need more phosphorus to support that increased biomass production. And a key point to finish is that our Australian soils are very old and weathered, and they're low in phosphorus. So this is really important. So our base storage of phosphorus is really low, which makes us very different to a lot of other countries. So when we talk about nutrient supply and release, it's on a different scale compared to what, say, North America is talking about. With very young, productive, fertile soils, it's more about how they can actually pull that phosphorus out of the system, whereas for us, our base amounts of phosphorus are just so much lower. So it's just a different world. So before we go any further, let's introduce the key players in our phosphorus cycling in soils. So first of all, let's talk about organic P. When we think about organic, we tend to talk about things of natural origin, derived from humus or from soil organic matter, kind of something that is not derived from chemicals. From a chemistry perspective, this basically just means that it's phosphorus compounds that contain carbon and oxygen. They're generally of plant or microbial origin, and they can't be generally taken up by plants. So it's a store of phosphorus in the soil that plants can't see. So as I mentioned, organic P actually refers to the chemical structure, with one of the key structures being things like inositol hexaphosphate, which I've shown here. Quite complex molecules, lots of carbon and oxygen in them. The next next player is what we call inorganic P. So these are phosphorus compounds that don't contain any carbon. These contain both plant available sources of P, such as superphosphate, MAP, DAP, etc., and insoluble sources. And shown in examples here, we've got one of the simplest phosphorus molecules around, which is orthophosphate, which is the key component in all our fertilisers. And then we have apatite, which is the main source of phosphorus, mineral phosphorus in our bones. Very complex structures. So organic P, inorganic P, purely about chemical structure. Plants see some of the inorganic P compounds. So then these two lots of compounds come together when it comes to our soils. You have our acid soils and our calcareous alkaline soils as kind of the two ends of the spectrum that are most P loving. They don't care if it's inorganic P or organic P, they'll hold on to both of them really strongly. And that's where a lot of our issues come from. And just some of the terms that we talk about when we think about phosphorus cycling is we talk about mineralisation, which is the loss of that carbon structure and the conversion of that organic P into inorganic P. Then from inorganic P, we move back to organic P through immobilisation, which is a microbially derived process generally. And then when we talk about these compounds and their interaction with soil, we talk about them becoming adsorbed or adsorption onto clay surfaces, or precipitation, which actually takes them out of solution and becomes a solid. So that's basically the language that we talk about with our phosphorus forms and interactions. So let's think about what happens when we add phosphorus to soil. We've got our P fertiliser, and let's say this is inorganic, so it's a superphosphate or an MAP. We apply that onto the soil. Most of it locks into the soil, and only a small amount of it is actually seen by the plant. Then when we talk about animal or plant phosphorus coming into the soil through manures or decaying plant matter, we've got biological breakdown of that material into organic P. That organic P can go straight onto the soil and get bound up, or it can get converted to inorganic P and get bound to the soil and locked up. And a small amount of that becomes available to the plant. When we get a lot of P bound up to soil, depending on what type of soil we're dealing with, that can also result in leaching to groundwaters or runoff to waterways, which is when our environmental risk with our nutrient strategies becomes important. So that's the big picture. So there's, as I said, there's 4 questions that we really need to be asking. Well, first of all, what do the different soil P tests mean? We talk about them a lot, but what are they? The second one is, what's the basis behind our phosphorus getting stuck onto soil, and why is that important? What do the microbes do in this system, and what can we do to change the availability of that phosphorus? So let's go through those. So our first question is, what are these different P tests and what do they mean? So I tend to find it's easier if you think about a bucket, and apologies for my poor graphics on this picture. I gave it a shot, but obviously graphic design is not my strong point. So there's 3 parameters that become important when we talk about soil phosphorus content, and together they can provide a clearer picture of our phosphorus nutrition. So first of all, these are soil Olsen P. Olsen P is something that's been around for a long time and is traditionally used in the pasture industry. So Olsen actually refers to the method of extraction, and actually the guy who first developed the method in the first place. So this method and this test gives an indication of the soil phosphorus that's immediately available for plant uptake. We call it an intensity measurement. So an analogy is how fast that phosphorus can flow out of the bucket. So if there's a hole, how quickly can it come out to provide phosphorus for that plant? Our soil Color P, also named for the gentleman who came up with the method, is a test that's traditionally used in the broader cropping industry that works on seasonal cycles. So this provides an indication of the amount of soil phosphorus which would become available to plants over that growing season. So an analogy might be how big that bucket of phosphorus is. And then our phosphorus buffering index, or our PBI, is a measure of how strongly that soil holds on to that phosphorus. So think about a sponge in the bucket, how much that sponge can suck up, and then what's left to then flow out of the hole. So the 4th thing that we think about is our total soil P. So that's all the phosphorus in our soils. Now this is rarely measured. Only in research purposes would we be measuring our total soil P, as most of that phosphorus in soil is tied up in mineral forms that aren't interacting with plants. So think about apatite, that really complex inorganic P structure I showed earlier. So to give some context, our coal P bucket may be between 1 and 10% of that total P in soil. So there's more there, we just can't see it. So that's why we tend to use those other measures. And I'd just like to go back a little bit more to that PBI method, which I understand is important with the work that's been done through the Uptake project. So just to reinforce, it's called the PBI, the Phosphorus Buffering Index, but we tend to talk about phosphorus buffering capacity, or how strongly that soil can hold on to phosphorus. Soil type is really important when it comes to looking at PBIs. So our clay soils are highly charged, they have a high chemical charge, and they will strongly hold on to that phosphorus. Whereas sandy soils have very low charge. So think about, obviously sand grains, very, very smooth, very non-interactive. They don't really bond well or chemically bind with other things really well, which is why soil structure is generally poor in our sandy soils. So it can't capture or hold on to that P. So there are some other factors that can determine the PBI, but clay content is generally the strongest driver. So increased clay content, increased PBI. So when we think about managing these 2 soil types, our clays and our sands, and think of loams and that in between, it's a very different strategy. So our clay soils need higher P additions, or larger amounts of P added to the soil, to bump up the amount of plant available P. So that means that there's enough P to satisfy that soil phosphorus binding, and there's still some left over for the plant. In comparison, the sandy soils need more drip feeding of P. So our low PBI, poor capture capacity, means that phosphorus can easily move through these soils and potentially into waterways or runoff, which can cause these environmental issues. When it comes to sandy soils, our soil organic matter is really important, as an increase in soil organic matter can increase that P capture through contributing to an increase in our PBI through both a chemical benefit through increased soil charge, which can hold more P, and biological benefit through increased soil biological function, which increases that cycling capacity of phosphorus and, of course, other nutrients. So how exactly does phosphorus get stuck in these acid soils? Well, we talked about the importance of pH and how our acid soils and our alkali soils, the extremes, most important when it comes to P capture. But when we look at that across the spectrum, and I don't think I'm not displaying this properly, apologies. But if we look at the soil pH on the x-axis down the bottom, percentage distribution on the y-axis, we can see that as our soil pHs change, the chemical forms of phosphorus in our soil also change. And when we get to these ends, our acid and alkali soils, that's when there's the smallest amount of phosphorus available for plants. And this amount can be from 0.1 to 15% of total soil P. So there's a lot there that the plant can't see because of that pH influence. So if we drill down a bit further into what is that pH influence, and if we were to put some acid soil under a very strong beam of light and actually seeing what was happening on the surfaces of that soil, we could see, eventually when it comes up, exactly how the different elements in soil are distributed, even at very small spatial resolution. And I'm just waiting. For it. So might just keep chatting for a bit. What we find is when we actually look at nutrients or even different elements in soil at, even at a micron scale resolution, we can see how it's not homogeneous in soil. So there's not like a blanket covering of phosphorus on all the clay particles. It's very much a spatially distributed arrangement. And what we can see is if we look at this spatial approach and then we drill down and put a very, very strong beam of light right into this heart of that phosphorus hotspot in soil, we can see from the spectrum, the X-ray spectrum that we generate, that we can say that phosphorus is actually strongly chemically bound with aluminium, which we would suspect based on the fact that aluminium is co-located with the phosphorus in that site. But this is very much a common phenomenon of that phosphorus aluminium binding. And then we can drill down to exactly how that binding occurs. But it just gives us an idea of what's happening at that small scale, so that when we're thinking about improving phosphorus availability and changing fertiliser formulations, etc., that we're actually doing it from a really strong chemical basis. So that was the chemistry side of what's happening. So what do the microbes do? So this is where, as I've mentioned, there's a lot of interest in understanding the role of microbes in our phosphorus supply. So we know that there's a range of P-related microbes in soil, and we know that when we have the breakdown of our large organic matter and manures, that the microbes produce enzymes called solubilisers that break that down into these specific organic P compounds. Then from that point, other microbes produce phosphatasers, which convert that organic P into inorganic P. And then other microbes come along and eat that inorganic P compounds and immobilise it within their microbial cells. So basically they use it to balance their diet. And so that means that this phosphorus is stored within the microbial cells and then only becomes available for re-release once those microbes die and the microbial biomass is broken down. And just on the right-hand side, just flagging that part of the diagram I showed earlier, showing how that organic and inorganic P conversion then also changes the P supply. Okay, so we know that there's key enzymes that microbes produce to change our phosphorus availability or to increase our P supply. So the next question is this P release related to a very specific microbial group, and can we then make use of that information in terms of nutrient availability? So the good news is there's been a lot of measurements done on the kinds of microbes that solubilise phosphorus and make it available for use. The bad news is that there's actually quite a lot of different microbes that do the same job. So this publication was done back in 2013 as a summary of what was known at the time, and the number of species has since continued to increase. So this is the range of bacteria, fungi, actinomyces, cyanobacteria and VAM that have a role in P solubilisation. So how do you pick a winner out of that? So that's the key, that's a very specific question around P solubilisation. But there's other roles that microbes play in increasing the availability of P in soil. So a key thing is through increased root growth, through extending P exploration, or the ability to find and scavenge for phosphorus in soil through VAM fungi. You can see the top picture of the VAM in soil, or through hormonal stimulation of root growth, branching or root hair development. You can see the picture of the root hairs. So basically, the more surface area you've got exploring into soil, the more chance that they can find phosphorus that's hidden throughout those soil layers. The other thing that you could do is look at is what they do is to change the P balance in soil through microbial consumption or eating that phosphorus and storing it in microbial biomass, which actually causes a diffusion gradient and causes more chemical release of phosphorus from soil surfaces into that soil solution. So we think of microbial biomass as like a slow release sink of plant available P, and full turnover of phosphorus in that system in biomass could be between 10 and 42 days. So if you've got a really strong microbial activity happening in your soil, the chances are that you've got ongoing cycling of that phosphorus and continual kind of a slow release drip feed system. The other thing that they do is that direct chemical solubilisation and mineralising of phosphorus from sparingly soluble forms. So these are from the inorganic P forms. The microbes can actually produce organic acids and other complexing or mineral dissolving compounds, which actually dissolve that inorganic P in soil. Or there's also, they can convert organic P forms into inorganic P through those extracellular phosphatase enzymes, which are also produced by plant roots. So let's go back to the whole microbial community thing, and can we actually kind of pick a winner as far as the kind of microbes go, and can we then produce some form of inoculant or something to add those microbes to soil to then increase the availability of P. So this has actually been looked at very closely over the years, to the point that the key performance of one of the really strong P solubilising bacteria, Penicillium biliari, was in looking at the performance of this as an inoculant on wheat crops across 47 different field experiments. And this was reported by Richardson and Simpson from SARO in 2011. Basically, they found no consistent benefit in terms of plant P nutrition and no relationship between the inoculation of using this P solubilising bacteria and any soil or environmental parameters, no relationship with growth, despite the fact that most of these trials were responsive to P addition. So basically what they're saying is that adding inoculants into dynamic competitive soil systems which have an existing microbial community has shown no consistent benefit. And the issue with that is that they can't compete with the native microbial community's home ground advantage. So the guys who are there first are there because they fit and they function well in those systems. The other thing to consider is that while the microbes may solubilise and release phosphorus for their own dietary needs, any subsequent benefits to plants may only occur following the turnover of the microbial biomass. So these guys aren't in it for P, they're just in it for themselves. And if there's any side effects that's great for plants, that's a bonus. But that's not what they're thinking when they rock up to microbe work in the morning. So inoculation of solubilising bacteria is unlikely to be a winner at this point in time, may change in the future. So what else can we do to change our phosphorus release in soil? So as I mentioned, homegrown advantage. So existing microbial populations have flourished in that soil because of their fit with that soil's chemical and physical properties. Microbial activity and community composition is also driven by interaction with various plant and microbially derived signal compounds and other regulatory compounds. So it's not all about P, as I mentioned before. It's just basically a side benefit. The other thing to consider is that microbes may also be limited in their P-related functions due to a lack of nutrients, the presence of pathogens, seasonal conditions, too much water, not enough water, too hot, too cold, or some other most limiting factor. So when we think about Liebig's law of the minimum, where's the lowest hole in the wine barrel that's going to cause inefficiency in that system? Okay, so it all looks pretty complex. All these things going on, don't really know what's going on. So now what do we do when we think about our P availability in soil, and how do we decide what to do? I know this is really corny. Basically, if you build it, they will come. So think of microbes as the residents of a house or the people who attend a fairground or something. It's got to be attractive for them to want to be there. So the ability of microbes, first of all, to be present in soil, to function well, and to release phosphorus is dependent on a number of factors. Key issue is soil pH, soil temperature, soil moisture, salinity, or not too much, a source of stored insoluble P. So basically you need some phosphorus in the system for them to work from. If your soil is completely deficient in phosphorus, your total P levels are really low, there's nothing there for the microbes to convert into plant available P. So they need something to work with. They need an energy source to keep them going. So that means maintaining organic carbon levels, looking at residue return in the soil, smart rotational grazing practices, maintaining strong perennial root systems, ensuring ground cover, all those things, and balancing your other nutrients. So we know that in our cropping systems, we need to ensure there's a bit more nitrogen in the system to be able to work through high stubble loadings without causing nitrogen drawdown. We know legumes in the system do a great job in capturing N from the atmosphere, which then creates that balance so that microbes can work more efficiently, or else use nitrogen fertilisers, etc. Basically work on the big things that may be most limiting in your soil system. So acid soils are a definite limiter. Most microbes can't function well in highly acidic systems. If you've got sodic or dispersive soils, you're likely to have low oxygen supply and also poor water infiltration. If you've got a dominance of weeds, that's not going to be very efficient in terms of how you can use any phosphorus that's solubilized. So looking at developing productive pastures with vigorous roots to capture and cycle phosphorus in the system through that ongoing organic deposition. So I just wanted to finally, before I finish, I just want to kind of go sideways just a little bit because I think this is really important. So how we measure and sample for our phosphorus. And I've spoken about acid soils a bit already. And I just wanted to leave this just before we finish by just flagging acidity tends to be a bigger problem than what we know. And a key issue with that is that most of the lime in our systems are broadcast onto our pastures or our no-till cropping systems. So we end up with a really strong lime layer in our surface, which can take years to move down the profile. But as you can see with that diagram that I've got, is that within that top 10 centimetres, your pH can change from very alkali or very satisfactory right down to be highly acidic by the time you get down to 10 centimetres. If there's a history of broadcasting and it's highly acidic, so that lime is being used up and not moving down the profile. So the problem with this is that when we take our soil sample, let's say down to 10 centimetres, and I'm sure there's different regional practices in terms of exactly what depths are used and how often they're done. But even across that 10 centimetres, you could have a really strong range of pH. But because that's all mixed together in the same bucket and sent to the lab, the pH comes back at 5.5, and you think, happy days, I'm doing pretty well. But you may still find that your microbial function is poor, your nutrient availability is poor, and you may not be able to grow some of the pulses or legumes that you think you should, or you're getting poor persistence of your subclovers or other legumes in your systems. So just have a think about that to see if broadcasting of lime and the lack of movement to depth could be an issue for you. The other issue is that with our modern cropping and pasture systems, our very dynamic and high biomass systems, we're exporting more alkali. We're also exporting more phosphorus through our grain, our hay, meat, wool and milk and other products. And hay is actually the biggest kicker in that. That's the one that exports most of our alkali or our high pH stuff from our systems. And of course, with use of urea or even with legumes, that can also contribute to acidity through end cycling processes. The other point to consider is that we're not sampling to depth like we should be in a lot of our systems. So below 10 centimetres, we could still be acidifying within our systems. So to look at doing some sampling to depth would be worth it. So just to flag, our P distribution follows similar patterns to our pH. And so you have a standard and a regular soil sampling program of our 0 to 10 or equivalent, and also include deeper samples to track pH at depth and also to track phosphorus movement at depth. Because if we're getting movement of P down, that could represent a system loss. So it's phosphorus that you're not using to grow plants, and also a potential environmental risk. So measure phosphorus and pH at surface and also at depth to make sure that you know what you're dealing with. So, sorry I had that little diversion. So basically, to summarise, what does this all mean? We know plant available P is only slowly available in our highly acidic or our alkaline soils. Our productive agricultural systems need P inputs through fertilisers or organic materials to replace that phosphorus that's removed by product. Okay, if we're exporting or we're removing food or fibre from our land, we need to replace the nutrients that we're exporting. If we focus the energy on managing the macro inputs on the farm scale, ensuring good ground cover, balanced nutrient requirements, apply lime or gypsum if needed, then the microscale residents, our microbes, will work. Many microbes contribute to increasing the pool of plant available P, and they also fulfil other roles in our soil systems. So I really encourage you to learn everything that you can around your soil. It's pH at surface and at depth. What's your PBI? What's your rooting depth? Is there any constraints to plant production? What's your clay and sand content, water dynamics, leaching potential, so that you can make responsible, informed decisions regarding your nutrient addition. So add what you need, but make sure you're not adding too much. So the other thing is that soil sampling is a relatively small cost compared to the cost of fertiliser or other inputs that you put in. So a consistent soil sampling strategy will ensure that you add as much fertiliser as you need, and also track your P usage to make sure that you're using what you're adding at most efficiently. So basically have a standard plan of sampling and do it the same way every time. So I think that's all I wanted to share with you tonight. I hope that made sense, and please let me know if there's anything that didn't. And thanks again for having me.
Thanks, Cassie. That was really illuminating, and I think as from a layman's point of view, I've got a much clearer picture, and I thought your buckets were great, by the way. And amazingly, I think you've explained it so well that nobody appears to have any questions for you. Well, I actually have a question. I guess there's a lot of talk about multi-species cover cropping these days, and given the diversity of microbes that can solubilize P, would it make sense then that if you build it, they will come? I'm assuming then that if you feed your native population with lots of diversity above ground, you're potentially going to grow that diversity below ground. Would that be a correct assumption, do you think?
Look, it's something that the science community is still very much on the fence. But from a fundamental basis, you'd think that if you've got more than one plant type, then you've got more than one root type, root architecture, root exudates, root exploration potential. So it would make sense to have a number of species in your pastures if possible. And some of even the old classics, so the grass and a clover mix, for example, work really well together. There is limited evidence of increasing the number of species has an additive effect. But I think the key would be, if you're going to increase your number of species, that you need to manage those species well. It tends to be that there'll be one or two species that will dominate, and over time everything else will wipe out. So I guess that the more species diversity you've got, the more structured and deliberate your management has to be above ground to make sure that you continue to get that benefit below ground. And make sure that your soil system is set up to support the plants that you want. For example, if it's highly acid, don't just go and put in a legume if it's not going to cope with those systems. Yeah. So it's definitely, it's a whole system approach of above, below ground, and what that soil environment's about.
Yeah. Now there is one question here. Is there a benefit in using liquid P?
I was actually expecting that question, knowing how common liquid P is in WA compared to other systems. So chemically, in most soils, there isn't any difference in its availability to the plant, unless you're on calcareous alkaline soils. That's the only soil type that's demonstrated an increase in the efficiency of P-release or availability. But I understand the liquid systems are becoming reasonably common in a lot of WA systems, and the availability of liquid P-fert. I guess in the, particularly in the lighter soils, a lot of the sands, I think liquids could work quite well, because you can apply those lower concentrations to add what you need, but not too much. Whereas that's a little bit harder in a granule system. But in more of a clay soil type, there would be no difference at all in terms of availability, because the actual compound of phosphorus that's in the liquids is actually the same as what's in the granules, and it will have the same chemical binding as soon as that liquid hits the soil. So it's really about what your systems are set up for, and efficiency of access, and being able to, I guess, co-apply that product with other products as well, being able to mix the tank. So chemically, no difference at all. So it very much comes down to what works well for your system.
Okay, well, there are no more questions at the moment, Cassie. So thank you very much. I know it's late there, so I hope you can stick around for the rest of the presentation. But I might just get you to turn off your microphone and your camera while I invite our other presenters on. If I can ask Rob to turn on his microphone and camera, and Kath, there we go. So we have Dr. Kath Lynch and Rob McFerrin from Department of Water and Environmental Regulation. Hi there.
Hi Kate, how you going?
Good. It's a bit weird, isn't it, when you can't see anyone? All right, let me get your presentation up.
Okay. And our camera's not opening your phone. That's on.
Okay, over to you two.
Okay, great. I think you're going to drive and I'm going to talk for a little while. Yeah, okay. Yeah. You've got the capable duo here. Thanks very much, Slough Blackwood, for giving us the opportunity to come and have a chat tonight, talk about something that we're pretty interested in, which is the Uptake project. I'd like to acknowledge our funders, which is the National Landcare Program and Healthy Estuaries WA, which is wouldn't be able to do this project without that funding. And I'd also really like to thank Cassie, who just did a fantastic intro for us and probably makes Rob's life particularly easy. So we thought we'd go to the pub, to be totally honest. So we'll just run through this quickly. So uptake, project aim, what we're trying to do is to help farmers improve nutrient efficiency on grazing farms in southwest WA to maximise both productivity and also to reduce nutrient loss to the environment. So I'm just going to do a little bit of intro of the project. It's got 3 different components: partnerships and extensions. So tonight's an opportunity to share some of that science with that extension side of things, innovation and fertiliser trials. Oh. So it's all right. So for me, the partnerships in this project is probably the thing that I'm most excited about. We've got a lot of people working together on this project, and I think that's really one of the most important things we can do. And I just want to acknowledge particularly our partners, the Department of Primary Industries and Regional Development, and I know some of them on the line today. We couldn't do this project without them. We have set up a technical reference group to support this project. There's about 25 people on that, and there's guys from the fertiliser companies, CSBP, Summit Landmark, grazing industry bodies, catchment groups, university researchers, I've forgotten anybody, and a couple of our Southwest farmers. And I'd like to acknowledge Tim Prosser, who's one of those farmers on that group, who makes a great contribution. We're also delivering the whole program through partnerships with catchment groups like the Lower Blackwood LCDC. And a critical partner in our program is the host farmers, who provide us with the land and the time and the opportunity to undertake these trials. Okay, so a little bit on the innovation side of the project. This is definitely worthwhile, a whole presentation on its own, but this is quite an exciting few gadgets that our friends in Deepert and Albany get to play with. So can I use this? Sorry. Okay, oops. So I'm not sure. That's all right. I'm just going to get Rob to drive. So the little yellow thing on the side is an X-ray fluorescence machine. It's currently used a lot in mining to look at the different elements in geology. So the guys are now looking at it to see, well, can we use something like this in place of soil testing to have a look at elements like phosphorus? So that's pretty exciting. Guys in Deepert are using the machine with lots of soil samples they've got in I was about to say cupboards, somewhere in cupboards in the back of the offices down there, and so we look forward to hearing that. The other part of the innovation is really looking at how to measure productivity quicker. So the guys who are doing the field work, it's really heavy labour-intensive work to get good productivity measurements, and I'm sure as farmers you would know that. And so the types of things we're looking at is like drone, and we've got a different pasture reader to try and take a little bit of that human labour out of it. And I think that's a really important part of the project, because what we're finding with the trials is there's lots more questions being asked by farmers and the people involved with the technical reference group. So we'll end up with more trials than we can probably handle. So a little bit of way to do that more efficient would be great.
Quick question, can people see the pointer? I'm not sure if it's selected or not selected. Oh.
Yeah, okay. We'll move on. Is anyone there? We'll move on. Okay, so the last component, which I'm just going to briefly discuss, is the fertiliser trials, and this is the big part of the project that we're working on. So we're aiming to do 36 fertiliser or phosphorus fertiliser trials across the southwest, from Peel Harvey catchment in the north down to the south coast Albany area. And on our map there, we've got a grey area, which is the sites that we've got in the Lower Blackwood and the Scott River. And I'd like to thank Brock Murray, Tim Crimp, who were in our last year's trials, and Andrew McNabb and Tim Prosser, who's in our Scott, that you hear a little bit more about tonight, being part of this project. And the overall aim of the fertiliser trials is to develop phosphorus response curves and determine critical values for southwest WA and compare them to national data. And I'm going to leave all that explanation of that up to Rob, although Cassie did a pretty good job in helping with us. But why this is really important, particularly these critical values, is this is the information that we use, or people who do the soil testing, like our colleagues at Deepert, or your fertiliser companies or your agronomists, to make their fertiliser decision recommendations. So a lot of people use a national data set called the Better Fertiliser Decisions for Pastures, and there's a table in that, and the maps that we do in the soil testing program that some of you might have been involved with. Those colours that you see in those maps, they're related to those numbers. So farmers have said to us, well, you know, how accurate are these numbers? How relevant are they to Southwest WA? They're a national data set. So we took that on board and thought that's a pretty reasonable question. And so what we're doing in this project is not only looking at to see how close those national numbers are, but we're actually really doing a whole new data set, which is pretty exciting in this space. So my last slide is the question is why we're doing the project. So at my end of the bargain is I deal with algal blooms and fish kills. It's not much fun, and they're mostly caused from nutrients, phosphorus and nitrogen getting into our waterways. The largest source of those nutrients come from grazing, but through most of the estuaries in Southwest WA. Soil testing programs in the last 10 years are showing that a lot of products have sufficient P, and a lot of farmers over the years have had to build up that P to get those good yields, but probably now have sufficient P in the soil. And what we're finding is we really want to build that confidence in farmers to have faith in that science, that the science through the soil testing is going to give them the right numbers to make fertiliser decisions to maximise productivity and profitability. So this project's about good science, hopefully inspiring farmers to have that confidence and an uptake in that science. Hand over.
Very good. Thanks, Kath. And also thanks, Joe and Joe Wren and Kate Tarrant from the Lower Blackwood catchment for the opportunity to present progress so far with some of the results that are uptake trial sites and some of the key messages we've observed so far. As mentioned, my name's Robert McFerrin. I'm the Project Uptake Coordinator. I think it's important to stress that I'm not a scientist. I'm a coordinator. I've also been a dairy farmer in the past and currently a beef producer. So fair to say I'm not the brains behind this project, and I rely heavily on scientists and agronomists involved with the project for when I'm preparing for field days and presentations such as this. People like Kath Lynch next to me, Dave Weaver, Graham Mussell, Luke Dawson from CSBP and Dave Rogers. Now, and because presentations aren't my specialty, we actually decided to put together a bit of a video which outlines the results today or to date so far for the Hardy catchment, while focusing on 2 sites. And I'd like to thank Luke Dawson in particular for his assistance in putting that together. But before we get to the video, I'd like to take a minute to explain some of the characteristics of nutrient response curves, and there'll be a bit of overlap with Cassie's presentation, so I apologise for perhaps going over some stuff that's already been discussed, but it's pretty important for the flow of the presentation and then moving into the video. So, yeah, so what's a nutrient response curve? Well, here's one. This is a generic response curve, or we could say it's a generic P response curve. It charts production on the y-axis over here, and against basically levels of P in the soil or applied to a pasture crop. Now you'll notice that's yield is expressed as a percentage of the maximum yield rather than absolute yield, and I'm not going to go into explaining the difference between those, or I'll get myself in a big knot, but suffice it to say it's expressed as a percentage of the maximum of what your soil type is basically capable of. So yeah, and we can basically see here that as the amount of soil P or P applied increases, so does the pasture production there. Yep, you'll notice that when it's first applied, it rapidly increases and it starts to sort of plateau out around here. Now, this point here, and we'll explain in further slides, is quite often referred to as a critical point, or I like to refer to it in farming talk as a target point for farmers to aim at, and I'll explain why in a moment. Also, this point here is quite important because it pretty much, or just above it, the 95%, it's the difference between what we would term in uptake as a responsive site and a non-responsive site. So you'll see in the video later on, we use the term responsive and non-responsive. So a responsive site, and hopefully this arrow is still working, a responsive site is in this part of the curve. So as we apply our known levels of P to the soil, we get a response. But once you pass this critical or target P point, you're not really getting a response from applying any more phosphorus. And we'll return to that in the video later on. So I'll just change the slide now. So back to these critical points. Now, depending on the farmer's desired level of production, just ignore that top curve for a moment. Let's just pretend this curve here is still a pasture response curve we were talking to earlier. Depending on the farmer's target, as I referred to, so if you're an intensive dairy farmer, you probably want to be producing at least 95% production. You want to be up around here. And if you're a sheep or beef producer, you may not need as much production. So your target or critical P point is probably around here somewhere, or 85. And you can see that has a big difference on the amount of P that's actually required in your soil. And so that comes back to the importance of doing soil testing and knowing what you're dealing with as a starting point and what you're aiming for over here in terms of pasture production. Now, nutrient response curves like these have been produced right across southern Australia over many years, and it's these response curves that help to form the basis of fertiliser application recommendations made by agronomists, and they're also included in the deeper soil testing program, which some farmers on here may be involved with. So once the farmer has completed his soil testing response, you can sort of determine, once you've got your soil test results, you can determine how much P basically you need to apply or we need to be aiming at. Now, unfortunately, as with life, it's not always as simple as it first looks. Certain soil characteristics, as was explained by Cassie, such as pH and PBI, can influence the availability of P in the soil. So, just change slide. And this graph, and now we focus on that extra graph that was in there. This graph illustrates the impact of PBI on the response curve. So, PBI, I don't know if I need to explain it again, but PBI, phosphorus buffering index, is basically the measure of the soil's ability to store phosphorus. And there are basically 8 different PBI groups for Western Australian soils, ranging from what we call a high PBI, and a high is anything over 280, and the range for PBI goes from basically 0 all the way up to 1,000. That's the measure. So there's 8 categories within the PBI range, going from, as I said, high, greater than 280, to exceedingly low, which is basically 5 or less. And it just so happens that the 2 sites that we're looking at in the video fall nicely. One is the very low PBI, or exceedingly low, so we're right at one end of the scale, and the other one's actually right over here in the other end of the scale. And you can see the difference that, or the effect. So a very low PBI doesn't take the addition of much P at all, and suddenly you're getting a very quick response. And so if we look at the actual coal well P of the 2 soils that we have, and you'll see in the video coming up, the coal well P for Andrew McNabb's site is 9, and so here's 20, so 9 would be about here. And the coal well P for Tim Pross's site is also pretty much the same, it's about 10. And you can see, because this is a response curve from a high PBI soil, the same level of P in the soil, you're going to get a very different response from applying any additional P if you are, in fact, low in P. If you're already high in P, well, you're not going to get the response at all there. So, so anyway, I think I probably missed a few things there, but looking up from my notes. But with that tutorial in mind, I don't know if I've helped you or hindered you, but we'll watch the video now, and hopefully that terminology will help with what you're about to see in the following video. Oh, here we go. We're away.
The Uptake project was established in 2019. The trials are triple replicated plot trials, plots 10 metres long.
Cool. Okay.
Thanks.
Each trial follows the same design of 5 different P rates with a nil fertiliser treatment included as a baseline treatment. All P rates receive an NKS basal treatment, as well as a nil P and highest rate of P having an additional treatment without the NKS basal to determine if there is any P response. The basal NKS treatment is applied after each cut and is providing luxury rates of N, K and S to ensure that the only variable to production is the different rates of P. The first site we're looking at is at Andrew McNabb's in Scott River. Andrew is a beef and sheep farmer, and his paddock has very low soil P levels in combination with very low PBI levels. The trial at Andrews was established in 2019 and continued in 2020 to determine not only the annual P requirements, but also to monitor the rate of P rundown over a 3-year period. What we've seen in this footage is a nil fertiliser treatment. We've applied no P, NK or S for the last 2 years. We can see compared to the application of 40 of P over the last 2 years that we are not seeing any visual or measured responses to P at this site. This footage shows that P is not limiting. However, with the addition of NKS basals, we're seeing a sharp increase in derived matter production to the tune of 3 tonnes per hectare, indicating that whilst P is not limiting production, another macronutrient, more than likely nitrogen, is limiting. As our rate of P increases with the addition of NKS, we are not seeing a significant increase in pasture production, further highlighting that P is not the limiting factor. In 2019, we saw no response to applied P at any rate. However, we saw a sharp increase in production with the addition of the basal NKS fertiliser. This year's accumulative results back up the findings from 2019 in that we are not seeing any response to P alone. However, there is a trend developing that with the addition of NKS basals, we're starting to see a P response. However, this is not yet significant, and the trial is still in progress for 2020 and hopefully into 2021 to further explore this trend. The second site we're looking at is roughly 10 kilometres away from the McNabb site, Tim Prosser's. This site has very different soil characteristics, with a very high PBI in combination with a low coal P level. As can be seen, there is limited pasture production with no fertiliser applied, as well as when we apply the NKS basals only. However, as soon as we apply the top rate of P with no NKS, which in this trial is 80 kilograms per hectare, we see a very sharp increase in pasture production, indicating that this site is very responsive to P. As we increase our P rates across this site, we can see a very visual as well as measured increase in pasture production. Another interesting observation at this site is that although we are not getting response to basal NK and S without P, as soon as we are removing the limitation of P, we're starting to get a synergistic effect of additional pasture production by adding the NKS basal to the tune of an extra 800 kilograms per hectare of production so far. This trial is still also in progress for 2020. Both of these trials highlight the need to soil test and identify the factors that are limiting pasture production. You then need to address the limiting nutrients in order of priority to ensure that only the correct nutrients are applied at the correct rates, to ensure that not only is money not wasted on applying nutrients that are not needed, but that those nutrients, such as P, are not being over-applied and potentially entering vulnerable waterways and catchments and contributing to environmental issues.
also, we missed out on the acknowledgements there. A big thanks to both Tim and Andrew. Kath mentioned earlier for making their properties available. So that gives you an idea of what we've seen from those two sites. It's also been, I think that's one of the strengths of the Uptake project, is the fact that we're going over 3 years. We can really monitor that rundown story and how different PBI soil types affect that rundown story. So we have Andrew McNabb's site, which is low PBI soil, and it's going. Hopefully, we can run that over 3 years and monitor that rundown story. We have another site here in Vass, the Grant Evans site. That's a higher PBI. It's not in the high PBI soil. It's in the mid sort of range, and that'll run for 3 years. That'll give us some important data on that rundown story on a different PBI soil type. And there's other things that we're monitoring with the uptake sites, like pasture composition and how that changes over time or throughout the growing season, but also with our continuing sites over a number of years, and the difference between ryegrass and clover with different fertilisers applied. So I'd encourage any farmers listening to keep looking out for details of any field walks or information coming out of these trial sites as we continue with the uptake project. As Luke mentioned in his at the end of the video, sort of real key messages that we've found so far is that, you know, it's the importance of soil testing. You've got to know what you're starting with, and you've got to know what you're aiming at, and base your decisions on the numbers. And also, yeah, definitely the undertake regular soil testing, because even I'm surprised at some of the early data out of year 2 of some of these trial sites. I think if you waited 3 or 4 years on some soil types, it might be a little bit too late in terms of that rundown or where you are in your soil P level. And yeah, most importantly, apply fertiliser to address limiting nutrients. Find out what's limiting first and work on that. And obviously apply it at the right time, and right location, and calibrate your equipment. So look, I'll leave it there. If there's any questions, I'm happy to take them. If they're above my pay grade or my understanding, I'll ask Kath or Graham to help me out.
Can you put on the website? I can't work the controls.
Oh, sorry. Yeah. Before I do that, there's a lot of partners involved in the Uptake project, and we're grateful for all their involvement, especially through the TRG or the Technical Reference Group, which is the group we established the trial design with, and it's an excellent collaboration. We're getting fantastic input from all those partners. So, any questions?
Well, again, you guys have obviously done a brilliant job at telling the story, because no one's asked any questions.
There
are definitely people watching. I can see their names, but maybe it might be a good time perhaps to hear from our two local farmers in question, Tim Prosser and Andrew McNabb. Perhaps if I can ask Tim and Andrew to turn on their microphones and their cameras so they can be revealed to the audience. That would be great. Are you there, boys? Oh, there's Andrew.
Hopefully.
And not sure where Tim is. Has he stepped out? Oh, there's Andrew. Excellent.
How you doing? Good.
I'm not sure where Tim is. Oh no, there he's coming in now. That's good. Hi, boys.
Yep.
Thanks for joining us. So perhaps you might like to tell us a little bit about why you got involved with Project Uptake in the beginning. Andrew, would you like to go first?
Yep. For us, it's just a opportunity to get a demonstration and get a better understanding of where our nutrient levels are.
Yep.
And for me, I'm born skeptic, so I like to see things proven on the ground. So it was a pretty good chance to get things, get into the group and get some proof on the ground for farmers to see.
Yeah. And have you found that the information that's coming out of the trial it's useful for you? And if it is, how will you use it?
I think it's only, it's still early days in the trial. Yeah, and it's probably the third year is going to be really interesting how the P rundowns are tracking.
Yeah.
I found it interesting because our ground's a lot heavier, just to see how much fertiliser is actually needed. It's yeah, quite surprising how much has to actually put on that heavy ground to get it to grow properly. The lighter stuff, which we have a lot similar to AJ's, that's always performed really good. But yeah, that heavier ground, it's surprising how much it does actually take.
Yeah. So have you got, I mean, as you say, you know, you still wanted to, there's another year to go yet, but are there any key messages for other farmers? I mean, what can other farmers gain from your experience with uptake?
I think identifying sites which have similar soil types to their own properties would be of use, and to follow that information.
Yeah, and to back that up with soil test too, the soil testing is the key to it all. But yeah, looking at these trial sites, it's sort of, if you can match those soil types up, it's sort of proof that the figures that we've been told all these years are actually true, and that they do mean what they mean, and you can be pretty relied on that they are correct.
Yeah, great. And are there any other comments you'd like to make in relation to the project, or in general, or about your experiences?
I think it's good to actually get these projects on the ground. It's good to get the money spent in the areas, and sort of we've always been told these things by departments, but very rarely do you actually get trial sites that farmers have access to. So I think it's a great initiative to actually get farmers trials on farms other than research stations that farmers have access to and can visit and have a look at to sort of see what is going on.
Yeah, right. Yeah.
Yeah, I think there's a lot of effort which has been put into doing these trials, and hopefully it's going to deliver good data from it, which is looking very good at this stage.
Yeah, no, really, really interesting. Well, it doesn't look like I've got anyone wanting to ask any questions, which is surprising considering such an interesting topic, but it's been very well explained by all the presenters. Someone's asked me if they can have the video link. That's fine. Just to let everyone know that this is being recorded, and I will be sending out the recording of the webinar next week for everybody, and the presentation. So don't worry about that. All right. Well, thank you very much to all our presenters, Andrew and Tim, Rob, Kath, and Cassie. It's been really, really informative and illuminating presentation this evening. And we look forward to hearing more about the test results next year. We'll probably do something, another round next year once we've got more results to speak about. So for everybody else, if I can ask you before you leave the room to complete our evaluation, that would be great. Thank you very much.
Thanks, Kate.
Thanks, everyone.
See you. Thank you, everybody.
See you.